stem cells and how stem cells are involved in regeneration. And of course, because the module is called signal transduction and oxidative stress and signal transduction, we are going to talk a little bit about the signal transduction mechanisms that are involved in the regeneration process. So, a short overview of this lecture. I will start by explaining you what regeneration is, what stem cells are. I hope this is a little bit something you know. And then afterwards we are going to dig in the signaling pathways of regeneration and how oxidative stress is involved in regeneration in cancer and how regeneration and cancer are actually linked. So regeneration, what is regeneration? In its most simple form, you can say that when one DNA strand is replicated in another, you have like a regeneration. Or when one cell forms another cell, you have regeneration. But in the biological form, we say that regeneration is when you have a new cell line or a new tissue, a new organ, or a new organism is formed. And actually the only organ that can regenerate in our body is the liver. But of course you have some forms or limited forms of regeneration in your skin, for example, or also at the level of your ribs. But in humans, the only organ that can regenerate completely is the liver. So regeneration actually is a fascinating thing. We don't understand everything because otherwise we can make new organs if one organ fails. And it is fascinating, and it's fascinating people since ages. Everybody knows, I think, the story of Prometheus, who was punished, and an eagle was eating its liver every day, and during the night the liver regenerates, and then afterwards, the day after, the eagle came back and ate the liver again. So this is the myth of Prometheus. But there are also myths that are happening during these days. I don't know. Yeah, these are the transplantations, or this one actually. But I don't know if you know this story. It's a story about a man. I think it was an American man who cut off his finger, or the fingertip, and what they did was actually make a powder of the bladder of a pig. And within this bladder there are stem cells, there are growth factors, and they say, or they pretend, that the finger grew back after bringing this powder of the bladder onto the cutted finger. So it was on, I think, on National Geographic, and there they explain it as a science, but we don't know if this is true. But we do know that when, for example, a child of, I think, beyond the age of 10 or 8 or 6 cuts off his finger or her finger, then it can regenerate the finger. But after the age of 10, we don't know. But maybe because of this growth factors or stem cells that are present in this bladder of the pig, it could be that some parts can regenerate. Nevertheless, several myths exist about regeneration, but what we do know is that there are some animals that can regenerate their body, or that can regenerate parts of their body. And actually these animals, when we invest these animals, then we can understand the molecular mechanisms of regeneration. And when we understand the molecular mechanisms of regeneration, we can manipulate them and maybe use them in our own research to, for example, generate new organs or new tissues that can be transplanted into humans when some organs fail. So what is regeneration? It's a perfect formation of new tissue when there's no scar tissue present. So it means that actually the whole tissue is renewed, is made again, but there's no scar tissue or no other impairments. This is possible because there are stem cells present. And in the human body, most the totipotent stem cells are in the embryo, but not in an adult body. But there are some animals, as you can see here, this newt. Everybody knows that when you cut off a limb of this newt, then he can regenerate his own limb. And actually, this is an organism that is studied very intensively, because if you know the underlying molecular mechanisms of this regeneration, then you can use it in regenerative medicine, for example, to make new organs. So stem cells are the underlying cells or the underlying mechanisms of this regeneration. And what is a stem cell? A stem cell is a cell that can renew, so that can make itself again and again and again and again, so that can renew unlimited. And a stem cell is a cell that can differentiate into another tissue. And this differentiation step depends actually if you have like a totipotent stem cell or a unipotent stem cell. So stem cells are classified according to their potency. A totipotent stem cell means that a stem cell can give rise to any other tissue. And a unipotent stem cell means that a stem cell can give rise to only one type of cell. And then, of course, you have some totipotent levels or pluripotent and multipotent levels in between. Multipotent, for example, means that a stem cell can give rise to different cell types which are linked together. So according to their potency, stem cells can be classified in the fact how they can differentiate into other tissues. When you have regeneration, you have a series of events that happens. First, of course, you have the amputation, so part of the body is removed. And afterwards, you have like a cellular migration of these stem cells, which are going to migrate to this wound, and they form like a wound cap. And this wound cap will form a blastema. And what is a blastema? A blastema is a cluster of cells, of stem cells, that can give rise to a new tissue, to different tissues, or to one kind of tissue, depending on the outgrowth that you want to form. So actually, when there is an amputation, you have a blastema that is formed. This blastema contains out of different forms of stem cells that can give rise to a different tissue, to a new tissue, to this outgrowth that you see here. I already talked about roots, and there are several other organisms that can regenerate. And depending on the organism, it can regenerate part of the body or its whole body. And of course, if you can have an organism that regenerates almost everything, that consists of out of totipotent stem cells, this organism can give you the most valuable insights if you want to perform regenerative medicine. Here you have like a lineage of all animals in the animal kingdom, and you can see that within these animals, the yellow ones, they can regenerate parts or whole organisms out of their stem cells. And you can see that from evolutionary point of view, that there is no point in the evolution where regeneration exists. So regeneration exists at different, or evaluates at different evolutionary points. So within the animal kingdom, you have like several organisms that can regenerate parts of their body or their whole body, as you can see here. Which are the most known model organisms for regeneration? Everybody knows the sea star, I think, which can regenerate one of their arms if this arm is amputated. I don't know if you know this hydra, which can regenerate by budding. So budding means that you have like a bubble coming out of the body and that it can regenerate actually a new hydra. You have like salamanders and zebrafish who can regenerate part of their body, who can regenerate like I don't know what you call them in English, doesn't matter. They can regenerate a limb, for example, or a fin, like what I was talking about. So, because they can regenerate parts of their body or their whole body, as the planarian worm can, they are very known and very used in this regeneration research. The organism we are using is the planarian. And actually, this is known because if you cut it, it can form a new animal, as you can see here. So if you cut this planarian, you can, in 3 parts. So you cut off the tail, the trunk, and the head. This will form these pieces. They will form 3 new animals. If you cut it in 278 pieces, you will have 278 new worms. So that's why this worm is commonly used in this regeneration research. It is very known because of this, because of its capacity to regenerate. This is a very unique property. So, of course, we are using it as a model organism. And why do we think, or why do we believe that this is a good model organism? Of course, because of its regeneration capacity. Because there are not that many species that can regenerate a whole new body. So for the relevance due to regeneration, of course, it's a really good model organism. But also, if you have like really small organisms, then you can manipulate them very easily. Like cell lines, cell cultures, you also can manipulate cell cultures and cell lines relatively easy, because if you put something in the cell medium, then it's taken up by the cells and you can inhibit something or upregulate something if you want. And that's the same thing you can do here also, because these organisms are very small. You can easily manipulate them, knock out, knock down some things, and study the mechanisms of regeneration. So, unfortunately, we have this worm, so not with the bright colors, because they can regenerate, but not that much. And this one is the only one, or one of the only ones who can regenerate everything. So, what does it look like, this worm? It's really cute. So here you see it swimming in the medium, and there's another worm, a scully actually.
And the knife comes and cuts it up.
And this is how they look or how they move. And why is it that they are interesting? How do they reproduce? What is their most unique property? You will see this here in this form. Okay. So here you see that this tissue is straightened, and you see the tissue remodeling occurring here. You have to wait a little bit, and then you will see the fragmentation and how this worm is cut into 2, and you will have two new worms. And this is the property which makes them unique and which makes them ideal to study regeneration. So here you see it will happen. Everybody ready?
Er waren bitterbeest hoor.
Will evaluate into a new worm. So we can do it manually. We can cut a worm if we want and study the process if we cut it ourselves, or they will do it themselves because this is the way that they reproduce. And of course, if you have a new model organism, you have to know a little bit about this model organism. You have to know that this model organism is not that huge at every time, because it's a top predator, which means that it's really aggressive when it comes to food. So these are small, call it small lobsters, and you will see, because of the fact that it is a top predator, it will actually, eat everything of this lobster. You will see now. So the platform has a mouth, and it comes out of the body, and then when the mouth comes out of the body, everything around it is eaten. So you see there will be nothing left from this small... Okay, so regeneration is a phenomenon that was studied since ages. It's really fascinating, but we don't know everything. Until now, we don't know everything. But as you can see here, this person, T.H. Morgan, already studied regeneration in 1800 or 1900, because it's at the end of the 18th century. And what does he want to do? We did know that these flatworms, that these planaria, can regenerate their body. But he didn't know how much. He did know that if you cut them in two, you have two new worms. But he had to know which body part can regenerate and which body part cannot regenerate. And the first experiment that he did to know the scope of this regeneration is actually, he set up an experiment, and here you can see it. He set up an experiment of this planarian worm, and he cut it in several parts. He cut it this way, and he cut it that way, and that he detected that every part that he cut could regenerate into a new worm. So no matter how he made his cuttings, every part could regenerate into a new worm. And these were actually the first experiments on this planaria. It's really basic, but these experiments showed that actually every part, except for a small part here, but almost every part can regenerate into a new organism. And this was, of course, really interesting phenomenon, but back then, he didn't know what the underlying mechanisms were. He thought it was really difficult to study this, so afterwards he wanted to study heredity, no regeneration, because it was more easy to study. But of course there are lots of other people who went to study this regeneration. And why do I think, or why do several other people also think that this is really interesting, is because these planaria can regenerate their heads, which means that they can regenerate their brains, they can regenerate their nerve system. And if you have like a model organism that can regenerate their nerve, that can regenerate their brain, then it can be very useful in neurodegenerative diseases like Alzheimer, like Parkinson, because if you induce Parkinson in these animals, they can evolve to a non-Parkinson organism. So if you induce the disease, they skip the disease, they remove it, and they come back. They become a healthy organism. So if you know everything on this regeneration, on this neuroregeneration, you can have new insights in manipulating applications or in the development of new drugs. So they can regenerate their head. They can regenerate almost all parts of their body. But how can a single cell, for example, also give rise to a new organism? Because that would mean that every stem cell in the body is pluripotent. If one cell can give rise to a new organism. This means that every stem cell is totipotent because this stem cell can give rise to a whole new organism. So this is a very recent science publication, and here they actually looked into this fact if one cell can or cannot regenerate into a whole new organism. And what they did was, I already told you that the underlying things of this regeneration are stem cells. So this worm, this planarian, is full of stem cells. And if you irradiate it, so if you irradiated this worm, then these stem cells are broken down, so the worm cannot live anymore, and it cannot, of course, not regenerate anymore. So what did they do? They irradiated a worm, and then afterwards they transplanted one new cell in this worm. And normally, if you irradiate the worm, it will die. But after this transplantation, you will see that from here out, new tissues are regenerated. Here you have this blastema formation, and from this point, a new worm was developed. So you transplant one cell, and from this cell, it regenerates actually a new worm, and everything from this worm, from here out, everything was renewed, every tissue was renewed, and after 60 days, you had a whole new organism which was healthy, and no effect of this irradiation was detected anymore. So this means that this one cell can give rise to any other cell that is needed in this organism, that is needed in this body. So this one cell is totipotent and can give rise to every tissue that you want. So of course, this is amazingly interesting, and you want to know the underlying mechanisms of this regeneration. You want to know which are the molecules, which genes are interested, which genes are involved in this regeneration process. I explained you earlier when I talked about model organisms that it was really easy to manipulate these planarian. Why? Because they are small. You can easily apply RNAi with these animals. Everybody knows what it is, RNAi, that you administer a new probe. This probe will degrade the gene of interest, and you have a knockdown of your gene of interest. And because it's easy to do this RNAi in our organisms, a whole RNAi screen was performed, which means that actually 200, 300, 400 genes were applied to the organism, and regeneration was followed. And if they detected that regeneration could not happen, we knew that this was a gene which was involved in regeneration. So we actually knocked down 400, 300 genes, and every phenotype which had no regeneration, had a gene of interest which is involved in this regeneration process. And how can you say that it has an effect on regeneration? As I told you earlier, when you irradiate your worms, when you irradiate your planarians, then you have then the stem cells will die, and you will have no regeneration. So you have a typical phenotype of irradiation, and the other genes or the other RNAi experiment was screened for this phenotype. And every organism who had this phenotype, then you knew that this gene was involved in regeneration because it had the same phenotype as the fact where we know the regeneration has stopped. So these are a few phenotypes of interesting genes. And what are these interesting genes? It are genes, of course, involved in signal transduction, and it are genes that are involved in cell cycle processes. Of course, we think or we hypothesize that also redox-related genes are involved in the regeneration, but there's not that much know about redox-related genes, but we will see a little bit about them in the end of the presentation. First, I want to talk a little bit about the cell cycle genes. And if you want to investigate cell cycle, you can investigate it, of course, at a molecular level. You can knock down several cell cycle genes and see what happens. But of course, you can also stain your cells and try to stain every cell which is in the cell cycle or which is in a certain phase of the cell cycle. And the interesting thing on in these organisms is that every dividing cell is a stem cell. So this means if you color a cell that is dividing, it's most certainly a stem cell. And at which phases of the cell cycle you can stain. You can stain, for example, in the mitosis phase, or you can stain here in the interphase. What is the underlying mechanism of the stains? Here in the mitosis, you can stain by an antibody, which binds to the H3 phosphorylation. So this is an H3 phosphorylation. It's formed here in the mitosis. So if you have an antibody which binds to this phosphorylation, you can stain every cell which is in mitosis. If you want to stain cells in the interphase, you can use another labeling method, which is BRDU. And what is BRDU? You actually add these BRDU molecules, and they are incorporated during DNA replication. So during the interphase, they are incorporated in the DNA strand. And of course, they are fluorescent. So if they are incorporated in the DNA strand, you can detect them under the fluorescent microscope. So here we have the BRDU staining of stem cells, and here we have an H3 staining of stem cells. So it means that every cell, which is at this time, which is in that part of the cell cycle, is stained. So this was a method this is a method that can be used to detect cell cycle events, and they see that, of course, when you irradiate these worms, then every stem cell disappears. So, based on this big RNAi screen, we detected some cell cycle related mechanism, which is of course logic because stem cells are dividing, but we detected also several signal transduction mechanisms. And we have seen that everybody knows this Wnt signaling is very, very important in the whole regeneration process. So this Wnt signaling, what is this Wnt signaling? Here you have a summary of most of the most known signaling pathways in the cell. And you see, or everybody knows, the MAP kinase pathway, or the RAS, or the apoptosis pathway. And here, in this part of the cell, you have the Wnt signaling. And as the other pathways, Wnt signaling means that you have a ligand, Wnt, which can convert a signal from outside of the cell to the cell nucleus, where transcription occurs. So as here, for the other pathways, Wnt also uses an external ligand that converts a signal from outside of the cell into the nucleus where transcriptional processes can occur. What are the known effects of this canonical Wnt signaling? Wnt is involved actually in the development of structures. So if one cell, a stem cell, has to form a new cell, it has to know where it has to go. If you have an embryo and every new tissue is developed, these cells have to know we have to go to the head, we have to go to the feet, we have to go to other parts of the body. It doesn't matter, but this body axis specification, this Wnt signaling is involved in telling the cells where they have to go. It's also involved in morphogenic signaling, but we will see this later. And wind signaling is detected, or one of the first times wind signaling was detected was in breast tumors. So we know that if this wind signaling is impaired, it can give rise to tumors or to cancer. It's also involved in stem cell differentiation, because if you have to direct cells, it are the stem cells that you have to direct, because if they come to the place where they have to be, there they will form new tissue. So it's involved in the differentiation process, but also in the patterning process to guide the cells to the exact place. So this wind pathway looks like this. You have here a receptor protein which is called bristles. And when the wind signal, or when the wind ligand binds to this bristle receptor, you will have an activation of this protein, the shackled. And the activation of this protein will inhibit this process, this complex. This complex exists of several proteins. And the function of these proteins in normal circumstances is the degradation of beta-catenin. But if the wind signal binds, this complex is inhibited, so the beta-catenin is not degraded anymore, which means that it can act as a transcription factor and stimulate the expression of genes. So if the wind ligand binds, you have beta-catenin, which migrates into the nucleus, binds to transcription factors, and actually stimulates or activates the transcription of several genes. And of course, if this process is disturbed, you have an overexpression of several genes, and that's where this beta-catenin and of course the wind signaling can act as an oncogene. Because if you have an unlimited expression, if the beta-catenin is stimulating the transcription factors continuously, then it means that you have a continuous expression of these target genes, which means they will let the cell divide and divide and divide or differentiate, and all the differentiation and dividing processes are disturbed. But beta-catenin is not only known here as it binds to or as it acts as some kind of transcription factor. It is also a known molecule of the cellular cytoskelette, where it acts together with, for example, actin, and it binds, it links cell to cells. So it links one cell to another cell, and there it's involved in the cell-to-cell communication. And here, it acts actually as a tumor suppressor. So when you look something up of beta-catenin, you will have to know that it's, on one hand, it's an oncogene, but on the other hand, it's like a tumor suppressor gene. And of course, we are interested in the involvement of this beta-catenin and of this wind signaling in regeneration. Because if you look in every kind of species, it doesn't matter which one, you will see that wind, which is green here in the figure, and beta-catenin, which is blue, exists over every organism. So this wind signaling pathway is a really, really, really conserved pathway. And that's why it's so interesting. It's interesting to know how it's activated and why it can lead the same pathway, why it can lead in one organism, it can lead to a whole other organism, it can regenerate everything, and in another organism it only can regenerate, for example, the liver. Why is that? What is this reason? What are the underlying mechanisms? And that is what we wanted to study. And the study that I want to explain now, you have to know a little bit more about this organism, and you have to know that there are different ways of cutting. Because to understand the next figures, you have to know that you can cut, for example, here, then you have only the head part. If you cut here, then you only have the tail, and this part is the trunk. So most of the time, if we talk about cutting experiments, you have three parts, the head, the trunk and the tail. If we talk about sides, then you have the ventral side, of course, and a dorsal side. So this is a study that looked into this, the role, the function of the beta-catenin in regeneration. And as you can see here, every ostrich is a new head. So this means that you have like a strange worm, a strange organism with 1, 2, 3, 4, 5 heads. A 5-headed organism, and this beta-catenin, as we know, is involved in the guidance of the cells to the exact place. And if this guidance is disturbed, then you have a strange phenotype. So this is just to show you how they have to look when nothing happens. And if we then administer beta-catenin to the trunk part, so to the middle part, then you see a 2-headed animal. So it means just by knocking down one gene, this trunk, this middle part, cannot develop a new organism. It develops a new organism, but with 2 heads. It's not with 1 head and 1 tail, but this trunk develops into a new organism with 2 heads, just by knocking down, not knocking out, by knocking down one gene. So here we knock down this beta-catenin, but what happens if we knock down a protein of the complex which is upstream of this beta-catenin? Then you have, as you see, also the trunk part, which forms a new worm, but with no head, or 2 tails. So if you knock down here, this complex, then you have an organism with 2 tails. If you knock down this beta-catenin, then you have an organism with 2 heads. So it means that just one gene can be responsible for this regeneration part of the head. And what does it mean actually? If you regenerate a head, then of course you have to regenerate everything which is in the head. You have to regenerate your nerve tissue, you have to regenerate your eyes, etc. And here you can see in these organisms, here, they stain the central nerve system. So in a normal worm, in a normal organism, here you have the central nerve system with the brains here. And when you knock down beta-catenin, the organism develops 2 brains. So it means that everything inside the brain, everything inside the 2 new heads is developed, 2 new brains. And here also the same, when you knock down this ABC complex, then 2 new tail parts are developed, also concerning the nerve system and other systems. So this was the fact when we have the trunk part, and the trunk part develops, when you knock down beta-catenin, the trunk part develops 2 heads. What about the head itself? It also develops when you let the head regenerate, and doesn't work, no matter, it doesn't matter because you see it here. Also the head part will develop a new head. What happens if you only have the tail part? If you only have the tail part and you knock down beta-catenin, what happens then? Then you will have an organism with multiple heads. Every protrusion here will form a new head. So if you have the tail part and you inhibit beta-catenin, it will develop not 1 head, but several new heads. And this is the same thing as you see here. Here also you have an intact organism which is knocked down with RNAi, beta-catenin, and you see these protrusions, these bubbles coming out of the body. All of these bodies will also form a new head by knocking down this beta-catenin. So if you take all these results together, it means that this beta-catenin, which is involved, or which is the downstream part of the Wnt signaling, is involved in this axis specification in the anterior or the posterior part or development of an organism. It happens in these organisms, but it also happens when you have an embryo and the embryo is developing a new child or a new baby, then it's also involved in this body axis specification, and you will see that when it's activated, then you will form the posterior part, and when it's not activated, so when it's inhibited, like in the knockdown animals, then it will develop the anterior part, it will develop the brains, the eyes, and the head of an organism. So this is the way they looked into the regeneration, and this is the way that they know that how the Wnt signaling and how the beta-catenin was involved in the regeneration, and of course in the making of a new animal. But how can one gene or one signal have a different effect on several cells? Because if you have an activation, then it has to activate all cells which are in the neighborhood. How does one cell know that it has to be an anterior cell, and the other cell that it has to be a posterior cell? It's because these Wnt ligands act as morphogenes. And what is a morphogene? It's actually, here you have the site of production of this morphogen, here, and of course, where it's produced, you have a high concentration of this morphogen, and when you go further and further, you have a low concentration of this morphogen. So let's say the Wnt signal is this morphogen. Every cell that lies close to the Wnt signal here has lots of this ligand available, and every cell which is more outside, which is more distant to the signal, has less ligands available to activate. And what does it mean if you have more or less ligands available? Then different genes can be activated. You can see here that when you have only one ligand that binds, you have only one gene, or gene A, which is transcribed. If you have 3 ligands that binds, you have another gene that is transcribed. So it's not only the signal that is important, but it's also the concentration of the ligand, the concentration of the signal that can differentiate in the transcription processes. This you can see here. Here you have the source of the ligand, source of the morphogen, and the cells which are really close to the source will have a strong activation of the signals. You can see here every ligand, every signal binds to a receptor, but the cells which are further away from this activation will have less ligand that binds, will have less signal, and here you can see 3 genes are transcribed, and here only 1 gene is transcribed. So this means that if you have more Wnt signal, you have more beta-catenin, so you have more genes that can be transcribed, and if you have less Wnt, less beta-catenin, and less genes that can be transcribed. This you can see here, where they actually did the same thing, but they gave lots and lots of RNAi, and they compared lots and lots of RNAi probes with only a small amount of RNAi probes, which means when you give lots and lots of RNAi probes, the gene is most certainly knocked down for not 100%, maybe 95%, but when you don't give that much RNAi probes, the gene will be knocked down, but only for a little bit. And what do we see when you knock down the gene for only a little bit? You have like another phenotype. You don't have a tail development, but you don't have 2 heads. And if you give more RNAi probe, so the gene is more and more knocked down, then you will have 2 heads or maybe 3 or 4 heads. So it means that it's not only depending on the type of gene, but also on the concentration of the gene. So processes or signal transduction processes are regulated, of course, by which gene that is activated or which gene is available, but also based on the amount of the gene. And this you can see in this experiment here. If you only have knock down a little bit, you have a little bit impairment, but if you knock down a lot, you have like multiple heads that arise. So you really have strong differences in phenotypes. And this you can see here in this graph. This is the dynamics of beta-catenin. So here, this is a normal regeneration, a normal phenotype. And you have beta-catenin, which is not that much present in the head, of course, but it's really high present in the tail. Because you know that if you knock it down, you have 2 heads. So it means that beta-catenin has to be limited to have head development. So in a normal, intact organism, you have a limited amount of beta-catenin in the head and a high amount in the tail. So what happens if you cut this animal? If you cut this animal here, it will regenerate the tail. And what is the underlying signal of this regeneration? This is an increase in beta-catenin. And this increase in beta-catenin will say to this organism, you have to renew your tail, you have to make your posterior back again based on this beta-catenin signal. What happens if we knock down this beta-catenin? This means that if you knock down this beta-catenin a little bit, you can see here, a little bit knocked down, it cannot make a tail again. If you knock it down a little bit more, it will develop 2 heads. And if you knock down it completely, it will develop multiple heads. So here you can see, based on the concentration of the beta-catenin here at the asterisk, if you have less beta-catenin, you will have more heads. So the amount of beta-catenin in the organism will tell you a little bit more about this differentiation process. And the same thing for this APC complex, which is the complex upstream of this beta-catenin. And here also the amount of this APC will tell you if this organism has to develop a head or a tail, and in this case, when it's impaired, it will develop 2 tails. Of course, you will have different Wnt ligands. So it's not only one Wnt ligand that binds to the receptor and that gives a signal. You have multiple ligands that bind and can give different signals. So you have the beta-catenin that is activated. The amount of beta-catenin that is activated depends on the amount of the Wnt ligands and, of course, on the type of the Wnt ligand. And if you have an organism, if you have a whole body, you have different Wnt ligands depending on what part of the body that you have. This was the fact, or this Wnt signaling is involved in the development of anterior-posterior development. But it's also involved in the ventral dorsal development. And here you can see if you inhibit this ventral dorsal signaling, then you will have the development of a new organism which develops on top of the other organism. So it means that here, at this side of myself, a new person will develop, like a Siamese twin. And this you can see here. So the dark part, or the more yellow part, is actually a new organism which is developed outside or on top of the other organism. Here you have 2 new eyes. These are old eyes from the first animal, and here you have 2 new eyes that are developed in the new organism, which grows on top of the other organism. So this means that the Wnt signaling is not only involved in the anterior-posterior development, but is also involved in the dorsal-ventral development via this BMP pathway signaling. I don't know if you've heard of this BMP pathway signaling, but it's a known pathway in bone development and to direct, of course, the bones where they have to grow. So there are growth factors, bone morphogenic growth factors that are involved in this signaling pathway. And then the last part of this signal transduction part, so you have the Wnt signaling that's involved by a beta-catenin in the anterior-posterior development. You have the BMP pathway, which is involved in the dorsal-ventral pathway. And here you have the NDK genes, Nodaraki genes, because this means in Japanese brain everywhere, because it were Japanese people which discovered these genes. And if you knock down these Nodaraki or these NDK genes, you will see that you have brain development in other parts of the body. But it only depends on which part of the organism that regenerates. So here, if you cut off this part, so if you cut off the white part and you let the red part regenerate, you will see that this regeneration will develop a new organism. Here the brains are stained, and you see that in the tail, new brains are developed. But if you do the same thing with the tail, everything is normal. And what is the underlying signal of this thing? It means that the Wnt signaling, as we have seen before, is interacting with this BA signaling, which is promoting this brain development. And this interaction will give rise to new brains via this NDK genes. So just so that you know, and just so that you see that there are several genes via this Wnt signaling that are involved in this anterior-posterior, ventral-dorsal, and in the brain development of these organs. So remember the RNAi screen, and when we knocked down like 300 genes, there were several genes, or most of the genes were involved in the cell cycle or in the signal transduction, mostly in the Wnt signaling. And by further studying these genes, we detected the way of regeneration and the involvement of beta-catenins. Of course, these redox-related genes are probably also involved in regeneration, but not that much is known about these redox-related genes in regeneration. What is known? For example, if you make a wound, then you have wound healing. And wound healing is actually some kind of regeneration. Because if I have a wound, I have to have new cells that make new skin, and hopefully I will not have a scar, and then you have a complete regeneration. Some mice can regenerate lots and lots of or big wounds, and we only can regenerate, of course, small wounds. But you have species, I don't know the name of the species, but you have 2 mice that can regenerate big parts of their skin. And they, of course, are used in the regeneration research also. But what do we know if you have inflammation that you have seen in the previous lecture of last week? You have ROS production. And these ROS, what do they do in this inflammation process? They actually are produced to kill bacteria, because if you have a wound, then lots and lots of viruses and bacteria come into this wound, and these ROS are produced as some kind of defense mechanism to kill the bacteria that come in this wound. But this ROS can also act as a growth factor, as some kind of signaling factor. And this signaling factor can stimulate cell proliferation, so can say to every cell, you have to come here because here the tissue has to be renewed. And one of these things that has to be renewed is the development of new blood vessels, of course. So this angiogenesis process is influenced by the ROS content and by ROS production. Also, the regrowth of the epithelia, the cell proliferation, is dependent of reactive oxygen species, and we don't know, but we hypothesize that this tissue remodeling is also activated by this reactive oxygen species. What do we know? That if you have a wound, you have to leave it open. You don't have to take, I don't know what you call it, bandages on your wounds, because wounds need oxygen. And why do wounds need oxygen? Because, as such, they can produce reactive oxygen species and they can renew their tissue. And several experiments have shown that if these wounds do not get enough oxygen, then you don't have tissue remodeling, which is good. You will have like scar formation or not a proper wound healing if you don't have enough oxygen. So you need oxygen to produce these reactive oxygen species, which can give rise to all these processes and which can co-regulate. Of course, they do not regulate everything. You also have growth factors, but they can co-regulate this wound healing process. And you have seen it in your journal club last week, which is one of the main producers of this reactive oxygen species, which are these noxes. And noxes are involved via calcium fluxes, for example, via the MAP kinase pathway in cell proliferation, in angiogenesis, and in fibrosis. And all these events will give you a new tissue and will give rise to tissue repair and to the wound healing. So we know that reactive oxygen species are involved in wound healing. We suspect that they are involved in regeneration, because wound healing is actually some kind of regeneration. But now the regeneration research is investigating the exact role of these reactive oxygen species and of course how they can communicate with, for example, the wind signaling to give rise to a proper regeneration process. When you put everything together, when you have like this signaling pathways and the redox-related pathways, you know that the underlying mechanism or the underlying cells of this regeneration are stem cells. Within the stem cells, several things happen. Here, the oxidative stress or the patterning pathways that are activated. We know that stem cells are involved in regeneration, and we also know that stem cells, undifferentiated cells, are involved in cancer. But regeneration, cancer, actually, it has a different outcome. But if you compare the underlying pathways, if you compare the signaling pathways and the redox-related pathways, we see that actually the regeneration process and the cancer process have links, several links, actually have lots and lots of links. The link is that here you have the regeneration pathway, and here you have the cancer pathway. And both pathways are known to have like extensive cell proliferation, dedifferentiation, and uncontrolled growth. And this last thing, that is the difference. So you have cell proliferation in both ways. You have cell migration in both pathways. The one thing that is different is that regeneration knows when it has to stop, and cancer doesn't. So, but if you study regeneration and if you study carcinogenic processes, you will see, and I actually started the presentation by telling you that the Wnt signal was discovered in a breast tumor. So regeneration pathways and cancer pathways are really linked and are really close to each other. The thing that is different is that you have cell proliferation here, you have abnormal cell proliferation in cancer. You have cell migration in regeneration, but you have abnormal cell migration in cancer. You have morphogenesis in regeneration, you have abnormal morphogenesis in cancer. But cancer can also mean that everything works fine until here and just not terminates. So actually, whole regeneration process goes on, but it cannot stop. And that's the difference between cancer and regeneration. But that's why organisms that can regenerate are also useful organisms to study cancer processes. Why is this? If you, for example, induce a tumor in this newt, what does this newt do? It actually amputates its limb, and it will grow a new limb. So this regeneration, organisms that can regenerate, when they develop a tumor, they can actually remove the tumor. So that's why these organisms are interesting, because they know how they can remove tumor formation. Because of the fact that this regeneration and this cancer pathway is linked, they know how to signal to the limb, just amputate the limb, make a new limb, and the tumor is removed.
Kan ik niet gaan oplossen?
This is actually seen also in a planarian worm, but another one that we use. And this worm is known because here's the anterior part, here's the posterior part. And if you induce a tumor in the anterior part, then you only have a small bubble, but not a tumor. Only have a little bit outgrowth, but not a tumor. But this part is able to regenerate. The posterior part actually is not able to regenerate. And if you induce a tumor here, then it really forms an aggressive tumor. So this is the proof that when an organism or a part of an organism can regenerate, it can remove a little bit of the tumor, but when it cannot regenerate, you really have an aggressive tumor formation, a strong tumor that forms in the non-regenerative part of the organism. So you have regeneration, you have cancer. Underlying to regeneration, underlying to cancer are the stem cells, and you have signal transduction pathways which are involved in cancer and regeneration via the stem cells. So these links makes or gives these organisms a potential to study the effect of carcinogenic substances, such as, for example, food or environmental factors or irradiation, other organisms that can give rise to cancer. And we use these organisms to study the development of cancer due to external substances. So how are these stem cells, the stem cells involved in the development of cancer via environmental factors? A stem cell is known because it's really, really protective. If you have a stem cell, it can take a lot. Stem cells are really resistant to stresses. It means that they have like a really good developed defense system against external stresses. So it means that when you have, for example, like reactive oxygen species, stem cells can defend themselves really quickly. If you study different kinds of tumors, you can see, and you will see this also in your research lecture on Wednesday, that this ROS signature can be used to discover or to detect this tumor. because some tumors are known to have high ROS levels, and some tumors are known to have low ROS levels. And it actually depends, or we think it depends, if these tumors came out of a stem cell or if these tumors came out of a dedifferentiation of tissue. So tumors that come out of stem cells really have, most of the time, have low ROS levels. And other kind of tumors that come out of differentiated tissue, they have really high levels of ROS. So based on this oxidative stress level, based on this ROS level, you can predict a little bit if this tumor comes out of a stem cell or actually develops out of a differentiated tissue. And we know that if a tumor comes out of this stem cell, we know that it's really chemoresistant, because stem cells really can cope with lots and lots of stresses, and that's why it's really interesting to study these stem cells under stress situations and to study these stem cells, how they can develop into a tumor, because they are really resistant. The tumors that come out of the stem cells, they are sometimes more aggressive. Unlike these stem cells, they are designed to cope with stresses. And stresses, like chemotherapy, of course, is also a kind of stress, and they are designed to cope with it and to grow no matter what. That's why it's interesting not only to study tumor formation as such, but to study stem cells under stress. And here I come back with the planarian again, because why it's useful, of course, to use these organisms to study these stem cells under stress, because they have lots of stem cells. These stem cells, as you know, are pluripotent, so they can give rise to everything, also to tumors, of course. And tumor formation in these animals can be linked to stem cells, because we know that every dividing cell is a stem cell, and we know that no other cell can be differentiated. So if you have a tumor in these organisms, it has to be a tumor that comes out of the stem cells. It cannot be a tumor that comes out of dedifferentiation, because we know that these cells cannot dedifferentiate. So the tumors that we study come out of the stem cell. And as such, we can investigate the underlying mechanisms of these carcinogenesis processes and develop new therapies against the processes that are activated. And what are these processes that are activated? Of course, several of the same processes as we had in the regeneration part. We use a carcinogenic substance, you have heard about it. We only use it because it's simple to administer, and you have tumor formation. It is known from literature that it is a strong carcinogenic element. And we see that after the administration of this carcinogenic element, we have a strong upregulation of HO proteins. HO proteins, everybody knows them. Defense proteins. And what was the most interesting results of this experiment was that when we administer the carcinogenic substance, we had this heat shock induction of every heat shock actually. But what was the most interesting thing? That these heat shocks were only present in the stem cell regions. So we know, based on this BRU stainings, we know where the stem cells are in organisms. We detected a strong induction of these heat shock proteins. And actually, we see that these heat shock proteins are located in these stem cells. Which means, because these are defense proteins, of course, that this can be an underlying mechanism of the stem cell defense, and this can be the fact why stem cells are that resistant against stresses. We know that this carcinogenic substance induces heat shock factors, We think it induces reactive oxygen species, and this is also true. As you can see, a strong decrease of the antioxidative defense means that you have less on the antioxidative defense, more reactive oxygen species. So next to this induction of neutral proteins, we also have ROS formation. So we have neutral protein activation, we have oxidative stress, so the organism is really under stress, and these reactive oxygen species will activate signaling pathways like the MAP kinase pathway. This you can see here, signaling pathways that are activated. Here, the ROS pathway, of course the cancer pathway, also located in the stem cells. So due to this stressor, we have oxidative stress. These reactive oxygen species activate signal transduction mechanisms among this ROS pathway, and both protected mechanisms as well as the cancer-related pathways are located in the stem cells. We have reactive oxygen species, we have the signal transduction, we also have, of course, apoptotic pathways. Because if you have tumor formation, if you have cell growth, you have cell division, and you have apoptosis. You have cell proliferation, you have cell death. And when this balance is disturbed, when there is more cell death, more cell division, less cell division, you can give rise to cancer. So we have seen that signal transduction pathways are involved. Now I'm going to let you see, of course, that apoptotic pathways are involved, also strongly induced, also present in the organism, but in the whole organism. So we have seen that the signaling pathways that are related to cancer, that are related to cell division, are only present in the stem cells, but the apoptotic pathways, they are present throughout the whole organism, which means that you have the cell proliferation in the active parts of the organism. This is to let you know, or to let you see that you, of course, also have DNA damage, improper formation of the different phases of the mitosis. And when we take everything together, we want to know, we already know that these reactive oxygen species give rise to cell proliferation via signaling via apoptosis. To prove this, we stimulated the cell cycle. This you can see here. And here in the staining, you can see an actual increase of stem cell production. Because every green dot is a stem cell, and when we induce or when we administer or carcinogenic substance, we have a strong increase in cell proliferation of these stem cells. An increase of cell proliferation can give rise to tumors. So when we administer the carcinogenic substance, we have an increase in cell proliferation. The cell proliferation, these are all stem cells that are proliferating, and these stem cells can give rise to a tumor afterwards. This you can see here, that every cell, this is a stem cell under control circumstances, this is a stem cell under stress circumstances. I don't know if you see a difference. I don't see a difference, which means that these stem cells are actually highly resistant due to these stressors. So you do have a strong increase in cell proliferation. You do have, you see it here also, you do have a strong increase in cell proliferation, but these cells are not affected at all. These cells can really, really cope with the stressor that is administered. So you have the cell proliferation. We already know what the underlying mechanisms are. If you link these underlying mechanisms all together. So you have the oxidative stress, you have the signaling, you have the heat shocks. If you link them together, this is a clustering analysis. If you link them together, you see that antioxidative genes, here this reductase and heat shock proteins are clustered together, which means that they are correlated. So when you have this condition of cell proliferation, you have three mechanisms that are activated: signal transduction, redox-related mechanisms, and the heat shock proteins. And we see that these heat shock proteins are linked together with the antioxidative defense. So it means that maybe these heat shock proteins are the protective mechanisms of these stem cells, which underlie the fact that they don't suffer from the stress, and that they give rise to cell proliferation, and that they give rise to a tumor. And this is the last experiment, what I'm going to explain you. We know that the stress factor induces cell proliferation. We want to know the underlying mechanisms. We did this screening, and we detected that the three processes, redox processes, signal transduction, and heat shock, were upregulated in the stem cells. We know that they are upregulated in the stem cells because we did this in situ hybridizations. You remember the stainings? We did a clustering analysis, which means that we linked redox-related genes with the heat shock proteins. That's why we decided to knock down the heat shock proteins and see actually if, when we knock down these heat shock proteins, if cell proliferation stops, and of course tumor formation will also stop. We knock down these heat shock proteins by soaking the animals in the probe, and then you can also inject it or inject it in the food. But we exposed our animals by soaking them in the probe. Then again, we performed a new qPCR screening. And what did we see? That when you knock down some of the heat shock proteins, cell division genes were also knocked down. So it means that when you knock down the heat shock proteins, you also knock down the cell division. When you knock down the heat shock proteins, you also knock down the signaling pathway, which is involved in tumor formation. So heat shock proteins protect these cells, and when they are removed or when they are knocked down, the cells or the stem cells cannot protect themselves anymore, and they cannot grow, they cannot divide, and signaling pathways are also impaired. Several stem cell related genes, known for the fact that they can stay to the stem cells, you have to differentiate or not, are also downregulated. And cell division, as you can see here, is also strongly downregulated. So by administering these heat shock proteins, or by knocking down these heat shock proteins in our organism, we can see that cell division stops and cell division is impaired. So we think and we hypothesize that heat shock proteins are involved and are involved in the cell division processes when a carcinogenic substance can give rise to cancer. And because these heat shock proteins are present in these stem cells are protected against external stresses, and they can divide without suffering from the stress. And this is also seen in therapies where they use chemo that works in the lipoprotein pathway. So they actually use, nowadays, they use chemo that works onto these lipoproteins, because these lipoproteins are very known protective mechanisms of these tumors, of these tumor cells, of this stem cell. So if we have therapies that knock these lesions down or that remove it, then you can cope or can attack the cancer. Of course, lipoproteins are very universal mechanisms, so they are not only present in the cancer cells, they are also present in every other cell. So we have to find, next to these lesions, we have to find, of course, a gene or a factor that is present only in the tumor cells, only in the cancer cells, and not in the rest of the body. Because if you, of course, administer the chemo to the patients, then if I take it orally, or if I take it by an infuse, then every cell of my body is exposed to this chemotherapy. And in every cell, this lipoprotein is present, so every cell will suffer from this treatment. So it's actually good treatment for the tumors, but a bad treatment for the rest of the body. So future research hopefully will lead to a new, more specific tumor-related issue. So this was the last slide of my presentation.
I'm sure that to meet more people.
So that's good. So you have the people of Maastricht have to go at the bottom of